Hello students, welcome back to Nucleus Neet Academy and I am Kiran Kumar, your biology NEET faculty. Today we are going to start a very interesting and important topic from chapter Biomolecules, that is carbohydrates. So I am going to teach you all the examples of carbohydrates with real life examples. It will be very amazing and interesting. Get ready with your pen and notebook. Now let us start carbohydrates. What are carbohydrates? In our day-to-day life, we will be using lot of materials which we will be simply aware of. They will be mostly carbohydrates. For example, early in the morning, we have sat before dining table to have delicious breakfast. The breakfast item, idli, dosa, roti, upma, all these are carbohydrates which are nothing but starch. When we eat starch, we will get energy, right? And starch is also present in the vegetable potato. Okay, so in the potato also there will be a carbohydrate called starch, and when we eat the starch we'll get the energy to do our work, right? So not only potato students, we can find carbohydrates in number of materials. And another example I can show you, tissue paper. After having our meal, we'll just clean our hands and mouth with this piece of material called tissue paper. It is also a carbohydrate. It is full of cellulose. Okay, so cellulose is also component of plant cell wall material. It is present in tissue paper, but we cannot eat the tissue paper. We cannot digest cellulose, but animals can digest cellulose. Another example for carbohydrate is cotton. Cotton is the purest form of cellulose. We use cotton for making diya wicks, okay, for lamp wicks, and also we use cotton for surgical purpose, for dressing wounds, etc. Okay, and also we make beautiful cloths out of cotton, which is also a carbohydrate. Okay, and another important carbohydrate I am going to show you, students, that is table sugar, which is very sweet to taste. We will use table sugar in sweets and different types of confectionery items. Look here, this is sugar. It is in the form of crystal. It is also a carbohydrate. Chemically, the table sugar which we use in day-to-day life in our juices and different other foodstuffs, sweets, etc. This is sucrose, also called cane sugar. It is obtained from sugar cane. And how it tastes, let us see. It is very sweet, very sweet. So we should not eat lot of sugar. Excess eating sugar will cause diabetes. It will cause diabetes. Okay, very sweet. So this is about an introduction to carbohydrates, and there will be lots of carbohydrates which we will be using in day-to-day life. I will tell you some more examples. In aloe vera leaf, if you open the aloe vera leaf, inside the aloe vera there will be slimy material called mucilage. Mucilage is also a kind of carbohydrate. And when we cut open lady's finger vegetables, in the lady's finger also you can find slimy gummy material which is mucilage. It is also a kind of carbohydrate. Okay, so carbohydrates are also present inside our body. They play very important roles in metabolic activities. We will discuss one by one in detail. Now let us see definition of carbohydrates. Carbohydrates are hydrates of carbon with general formula CnH2nOn. So there is carbon, hydrogen and oxygen. There are three elements in the structure of carbohydrates, and they are largely made up of three elements: carbon, hydrogen and oxygen. Okay, now let us see another definition of carbohydrates. Carbohydrates are polyhydroxylic. They are polyhydroxylic aldehydes, or they can be polyhydroxylic ketones, and their derivatives are polymers. They can be their derivatives or polymers. What are polymers? Large molecules with high molecular weight which are made up of small repeating units called as monomers or called as polymers. Okay, so this is about the second definition of carbohydrates. Now carbohydrates can be classified into two types depending upon the groups which they have. Carbohydrates with aldehyde group, CHO group, are called as aldoses. Similarly, carbohydrates with ketone group are called as ketoses. So there are two types of carbohydrates based upon the groups. They are divided into two types. Those carbohydrates which have aldehyde group are called aldoses. The carbohydrates which have ketone group are called as ketoses. Okay. Now let us see some examples of carbohydrates. Starch. Just now we have discussed about starch. It is one of the important carbohydrate which is seen in plants. So plants will store their food material, their reserve food material in the form of starch. Okay, it is the reserve food material in plants, and we human beings, we eat lot of starch on daily basis in order to get energy. Starch is present in food grains such as rice, wheat, and it is also present in potato. Okay, next is glycogen. Glycogen is also called as animal starch. It is reserve food material in animals. It is stored in organs such as muscles and livers in animals. Okay. Next is inulin. Inulin is also a storage reserve food material which is seen in some plants such as garlic, onion, and artichoke plants. Okay. Human beings cannot digest inulin, this carbohydrate. Okay. Next is cellulose. Cellulose is completely different. The function is different. Cellulose is the structural polysaccharide. That means it provides mechanical support to the plant cells in the form of cell wall. So cellulose, it forms the primary cell wall material of plants. It makes the rigid cell wall of plants. Okay, that is about cellulose. Cellulose is present in cotton. Okay, we cannot digest cellulose because human body does not have enzymes which can break down cellulose. Next is pectin. Pectin is also a cell wall material seen in the plant cells. Next, chitin. Chitin is the cell wall material which is seen in fungi, and it also is present as exoskeleton material in insects like dragonflies, cockroaches, beetles, etc. Heparin. Heparin is an anticoagulant. This material is present in the blood of animals. It prevents coagulation of blood. Heparin. Next is mucilage. Just now we have discussed mucilage is a slimy substance which is having sticky nature. Mucilage, it is produced by both plants and animals. In plants you can see mucilage in aloe vera leaf. Just now we have discussed. And also it is also present in lady's finger fruit also. Mucilage. It is a slimy material. Okay. This is about list of polysaccharides. So whatever I have wrote in this line, all these are polysaccharide carbohydrates which are essentially large molecules with high molecular weight. Their molecular weight will be above 10,000 daltons, nearly above 10,000 daltons. They are high molecular weight carbohydrates. They are complex. Okay. Simple carbohydrates, we are going to discuss glucose. Glucose, it is the most common simple carbohydrate which you can see in all the living cells. The cells will use glucose as a respiratory substrate in order to produce ATP energy. Next is fructose. It is called fruit sugar. It is present in fruits, in all the fruits. Galactose. It is present in many living organisms like peas, beans, etc. So it is also component of milk sugar. It is also present in the milk sugar, galactose. Sucrose. Sucrose is another sugar which is available in large quantities in sugar cane. So the sugar cane has sucrose. The sucrose can be also called as cane sugar. It can be also called as cane sugar. The table sugar which we use in our house in day-to-day life is nothing but sucrose, cane sugar. Next is lactose. The name itself tells lacta. Lacta means milk. It is present in the milk. It is called milk sugar. It is present in milk. Next is maltose. Maltose is also called as malt sugar. It is present in the cereals such as barley, rice, wheat. During germination of the cereal seeds, these seeds will produce the sugar called as maltose. Hence, it is also called as malt sugar. Okay. So these are some of the carbohydrates. Now let us see the flow chart of carbohydrates we will draw in a moment. Students, now let us see classification of carbohydrates. Carbohydrates can be classified into three groups based upon their complexity. The most simple type of carbohydrates are monosaccharides, little bit complex carbohydrates are oligosaccharides, and the most complex carbohydrates are polysaccharides. They have highest molecular weight of about 10,000 daltons. Okay. Monosaccharides, as the name itself tells, mono means one. So monosaccharides, these are the carbohydrates which are made up of only one or a single sugar or monomer that cannot be hydrolyzed any further. Okay, and they are simplest form of sugars. They cannot be hydrolyzed any further, and they are sweet to taste. They are crystalline in nature and they are soluble in water. So these are the properties of monosaccharides. And just now we have discussed a few examples of monosaccharides. Your glucose is a monosaccharide, and also fructose is also a monosaccharide, and also galactose is also a monosaccharide. They are all sweet sugars. Now, based upon the number of carbon atoms, based on number of carbon atoms, based on number of carbon atoms present in the monosaccharides, they can be further classified into 1, 2, 3, 4, 5 categories. Okay, the monosaccharides which have 3 carbons are called as trioses. They are called as trioses. They have general formula C3H6O3. This is the general formula of trioses. Look here, there are 3 carbons and double amount of hydrogens, again 3 oxygens. Okay. Next is tetroses. Tetroses have 4 carbons. Tetroses will have general formula, come on students, C4H8O4. These are tetroses. The 5 carbon monosaccharides are called as pentoses. Pentoses have general formula C5H10O5. Pentoses. Next is 6 carbon monosaccharides are called hexoses. They have general formula C6H12O6. Okay, so this is very familiar, right? C6H12O6, formula of glucose. Not only it is a formula of glucose, it is even the formula of your galactose and also your fructose. Okay. Next is 7 carbon monosaccharide are called heptoses. Heptoses will have general formula C7H14O7. This is the general formula of heptoses. So this is about an introduction to monosaccharides. Now let us see examples of these different types of monosaccharides. Trioses. Example of triose is glyceraldehyde. Glyceraldehyde is a 3 carbon sugar. Example of a tetrose with 4 carbons is erythrose. Erythrose. Example of pentose is ribose. It is present in nucleic acids. Hexose example, the most familiar, glucose. An example of heptose, pseudoheptulose. Pseudoheptulose. So these are the examples of different sugars. Okay, glyceraldehyde, erythrose, ribose, glucose, and pseudoheptulose. Now let us see oligosaccharides. What are oligosaccharides? Oligo means few. So oligosaccharides are carbohydrates which are made up of few monosaccharides as monomers. They are carbohydrates which are made up of about 2 to 10 monomers of monosaccharides. So these monosaccharides, they will be repeating in 2 times or 3 times. Next, what are oligosaccharides? Oligo means few. They are made up of few monosaccharides or monomers. They are made up of about 2 to 10 monomers. So those carbohydrates which are having 2 monosaccharides attached to each other. Each monosaccharide can be called as monomer. Okay, one monomer is attached to another monomer, then it gives rise to oligosaccharide, which is called disaccharide. Disaccharide. What is a disaccharide? A disaccharide is a carbohydrate which is made up of two monomers or monosaccharides which are attached to each other by a bond called glycosidic bond. Okay, they are called as disaccharides. Examples of disaccharides are sucrose, which is your cane sugar, and also your lactose, which is milk sugar, and maltose, which is a malt sugar. These are the examples of disaccharides. Now oligosaccharides, which are made up of three monomers or monosaccharides which are held to each other by two glycosidic bonds, we call them as trisaccharides. Look here, there are three monomers or monosaccharides attached to each other by glycosidic bonds. They are called trisaccharides. Trisaccharides. They are called trisaccharides. Example of trisaccharide is raffinose. Raffinose. Next, the oligosaccharides, which are made up of four monomers attached to each other by three glycosidic bonds, they are called as tetrasaccharides. Tetrasaccharides. Tetrasaccharide example, stachyose. Okay, next, the oligosaccharides which are having five monomers attached to each other by glycosidic bonds, 1 2 3 4 5 monomers are held to each other by four glycosidic bonds. We call this type of molecules as pentasaccharides. Pentasaccharides. Example of a pentasaccharide is verbescose. So these are the different types of oligosaccharides. Can you tell which is the most complex oligosaccharide over here? Exactly, that is verbescose, which is having 5 monosaccharide units in it. Okay, this is the simplest oligosaccharide here. Simplest oligosaccharide, right? It is the disaccharide which is having just only two monomers. Sucrose, lactose, maltose, all these are disaccharides. They are all sweet to taste. Then what are the properties of oligosaccharides? They have the same type of properties of monosaccharides. Just like monosaccharides, they are sweet to taste. They are sweet to taste and they are crystalline in nature. They are crystalline in nature, they form crystals, and they are soluble in water. They are soluble in water. Okay, but they can be hydrolyzed into simple sugars which are called as monosaccharides. So when we hydrolyze disaccharides, we will get 2 monosaccharide units. So when we hydrolyze trisaccharides, when we cut these bonds, then we get 3 monosaccharides. Hydrolysis of tetrasaccharides will give 4 monosaccharides. When we hydrolyze pentasaccharides, how many monosaccharides will get? 1, 2, 3, 4, 5. 5 individual monosaccharides we are going to get. Okay, understood the concept, students? Oligosaccharides are also sweet to taste. They are crystalline in nature. They are soluble in water. Here monosaccharides, they cannot be hydrolyzed any further, but our oligosaccharides, they can be hydrolyzed. Okay, so this is the difference. Now let us see polysaccharides. Polysaccharides are also called glycans. They are also called glycans. They have high molecular weight above 10,000 Daltons. They are large molecules. They are large molecules made up of several monomers. The monomers can be any type of monosaccharide. It can be any type of monosaccharide. These molecules are insoluble in water and they are not sweet to taste and they does not have crystal nature. And the polysaccharides, based upon the type of monomers which are present in their structure, they can be divided into two types. Homopolysaccharides. Homopolysaccharides. So they are made up of a single type of monomer. They are made up of single type of monomer. Okay, next, second type of polysaccharides. Those polysaccharides which are made of more than one type of monomers, they are called as heteropolysaccharides. They are called heteropolysaccharides. Heteropolysaccharides, they are made up of more than one type of monomer. Okay, now let us see examples of homopolysaccharides which are made up of a single type of monomer. So examples: starch. Starch is homopolymer of glucose. Glucose monosaccharide, which acts as a monomer, will be repeating several times to form a large chain-like structures. Ultimately, it forms a molecule called as starch. Similarly, another molecule, glycogen. Glycogen is also a homopolymer of glucose. Okay. Next is inulin. Cellulose. Cellulose is also a homopolymer of glucose, but its role is different. It has a structural role. It is not a reserve food material. Next, chitin. Chitin is also a homopolymer. It is present in the cell wall of fungus and also in exoskeleton of insects. These are all homopolysaccharides. Next, heteropolysaccharides. Examples of heteropolysaccharides. Pectin. It is cell wall material which is seen in the plants. Pectin. Next is peptidoglycan. It is cell wall of bacteria. Peptidoglycan. It is the cell wall material which is seen in the bacteria. Peptidoglycan. Next is mucilage. Mucilage, the slimy material in aloe vera, in lady's finger, mucilage. It is also a heteropolysaccharide. Next is heparin, anticoagulant in blood, heparin. Next is hyaluronic acid. Hyaluronic acid. Hyaluronic acid is another heteropolysaccharide which is seen in animals. It is present in the synovial fluid of joints and also it is present inside the eyeball. Okay. Hyaluronic acid, it is having lubricating properties in the body of animals. Hyaluronic acid. Next is agar. It is present in algae. It is a phycocolloid. It is present in the algae, agar. Next is chondriatin. Chondriatin sulfate. Chondriatin sulfate, which is present in cartilage. So these are all the examples of heteropolysaccharides. Okay students, copy down. Students, now let us see structures of some monosaccharides. We will discuss hexoses first. What are hexoses? Hexoses are monosaccharides which are made up of 6 carbon atoms. Okay, there will be 6 carbon atoms in the structure of hexoses and they have the general formula C6H12O6. C6H12O6. What are examples of hexoses? Your glucose, galactose and fructose. All these 3 molecules have the same formula C6H12O6. Okay, so now first we will start with glucose. Glucose is having 6 carbons arranged in the form of a ring structure. This ring structure is called as pyran ring. It is called pyrene ring. So here you can see this is the first carbon, second carbon, third carbon, fourth carbon, fifth carbon and oxygen. So total how many members are there in this ring? There are total 6 members in the ring. 1, 2, 3, 4, 5 and 6. So a pyrene ring is a 6-membered ring. It is a 6-membered ring. Okay, so your glucose is having pyrene ring structure. It is having pyrene ring structure. Now look here, the first carbon is held with second carbon by a bond. Second carbon is connected to third carbon by a bond. Third carbon with fourth, fourth with fifth, and fifth is attached to oxygen. This forms a pyrene ring structure. Attached to the first carbon there is OH group, H. Second carbon OH group, hydrogen here. Third carbon is having OH group towards top, hydrogen down. Fourth carbon OH is facing down, hydrogen is upside, and fifth carbon it is attached to the sixth carbon. So note here, the sixth carbon is out of the pyrene ring structure. The sixth carbon is attached to OH group. So this forms the structure of your glucose. Okay. Now galactose. Galactose almost the same structure, but there is a small difference. Can you recognize? Yes. Look here, on the fourth carbon the OH group is facing up, but here in glucose the OH group is facing down. So this is the only difference which you can see in the galactose, and formula is same. Next hexose is fructose. Fructose is also called fruit sugar. It is also called fruit sugar. It is abundantly present in all ripened fruits which we eat. Okay. So in the fructose ring structure you can see there are first carbon, second carbon here, third carbon, fourth carbon will be here, fifth here, and sixth carbon is out of the ring, and the fifth carbon is attached to oxygen. In this ring there are how many members? Look here students, 1, 2, 3, 4, 5 and 6. There are 6 members. 1, 2, 3, 4, 5. I'm sorry, there are only 5 members in the ring structure. So this is called furan ring. This is called furan ring structure. A furan ring structure, it is a 5-membered ring. It is a 5-membered ring. So fructose have 5-membered ring which is called furan ring. Okay, so look here the members, 1, 2, 3, 4, 5. Only 5 members are there in the ring structure. And where is the first and sixth carbon? The first and sixth carbon are positioned out of the ring. So you can see here the first carbon is attached to second carbon over here, and your sixth carbon is attached to the fifth carbon over here. Understood? So in the second carbon CH2OH is present down, on fifth carbon CH2OH is present towards up. So this is the fructose furan structure. Understood students? Copy down these 3 structures in your notebook. Students, now let us see structures of some 5 carbon sugars which are called as pentoses. Pentoses are monosaccharides which have 5 carbons. They have the formula C5H10O5. C5H10O5 is the general formula of pentoses. Examples of pentoses is ribose sugar and deoxyribose sugar. These 2 sugars are very important. They are components of nucleic acids. They will be present in nucleic acids, that is DNA and RNA. First let us study the structure of ribose sugar, the pentose sugar. In ribose sugar you can see there are 5 carbons. Here will be first carbon, second, third, fourth and fifth carbon. Total there will be 5 carbons. They are forming a ring-like structure. What type of ring is this, students? This is a furan ring. What is this ring? This is a furan ring which is a 5-membered ring. So in this furan ring, on the first carbon OH is facing down, second carbon down, third carbon down, and fourth carbon is attached to the fifth carbon, CH2OH. Okay, fourth carbon is connected to fifth carbon. The fifth carbon is outside the furan ring structure. So this is the structure of ribose. Ribose sugar, it is present in RNA. It is present in which nucleic acid? It is present in RNA. What is the full form of RNA, students? Ribonucleic acid. What is full form of RNA? Ribonucleic acid. So ribose is component of RNA. Okay. Next, coming to deoxyribose. So it is another type of pentose sugar which is almost similar to our ribose, but there is a small difference. Look here, on the second carbon, you can see here the OH group is absent. OH group is absent. On both sides there is hydrogen. Okay, so one OH group is absent. So one oxygen atom is absent. That's why we can call it as deoxyribose. Deoxyribose. One oxygen is missing in the structure of ribose. On second carbon atom, it is called deoxyribose. It is also having furan ring. It is also having furan ring structure, and it is present in DNA. This molecule is present in DNA. What is full form of DNA, students? Deoxyribonucleic acid. So your DNA is made up of this molecule. It is one of the molecule which is present in DNA. Understood? Copy down, students. Students, now let us discuss about disaccharides. What are disaccharides? Disaccharides are carbohydrates which are made up of only two monosaccharides. One monosaccharide is attached to another monosaccharide by a bond called as glycosidic bond. The bond is called as glycosidic bond. Let us see some of the examples. The first example is maltose. Maltose is also called malt sugar. Malt sugar, it is present in the germinating seeds. If you observe the structure of maltose, there will be two monosaccharides, alpha glucose, alpha glucose. So look here, each alpha glucose, it is a 6 carbon monosaccharide. So this glucose is going to bind with another glucose by a bond called glycosidic bond. So look here, this is the first carbon of our alpha glucose. This is the fourth carbon of another alpha glucose. So these first and fourth carbons are going to interact in bond formation. Look here, both of them have OH groups. When these two molecules come close enough, then what happens? A water molecule will be removed. A water molecule will be removed. Okay, after removal of water molecule, what is going to happen, students? Look here, there is a oxygen, a lone oxygen is present. So H and OH are gone in the form of our water molecule. This oxygen, it will be shared between first and fourth carbon atoms. First and fourth carbon atoms of two adjacent glucose molecules. This results in formation of a bond. This bond is called glycosidic bond. What is that bond called? Come on, tell students. Glycosidic bond. Understood now? Understood now, students? So this bond, we are calling it as glycosidic bond. It always involves removal of water molecule. Okay. So I will ask you a simple question. If you want to break this maltose molecule, you should break which bond? You are right, you have to break the glycosidic bond. In order to break the glycosidic bond, again you should add the water molecule. So when you add the water molecule, automatically the bond gets broken and two glucose molecules get separated from each other. So breaking of any type of carbohydrate which involves breaking of glycosidic bond is called hydrolysis. So breaking of glycosidic bond is called hydrolysis. Okay, lysis means breakdown. So when water molecule is added, then only we can break the glycosidic bond. Hydrolysis of maltose yields which sugars? Can you tell, students? Hydrolysis of maltose yields 2 glucose sugars. Understood the concept? Now let us see some more examples of disaccharides. First disaccharide we have observed, maltose. Maltose is made up of 2 glucose molecules which are held by alpha 1 to 4 glycosidic bond. What is alpha here? Alpha represents the type of glucose. One alpha glucose attached to another alpha glucose. That's why alpha 1 to 4 glycosidic bond. 1 represents first carbon, 4 represents fourth carbon. Okay. Similarly, lactose. Lactose is called milk sugar. It is present in the milk. Lactose is made up of 2 monosaccharides, galactose and glucose, held by beta 1 to 4 glycosidic bond. Next, sucrose, also called cane sugar. It is present in sugar cane. We use sucrose in the form of table sugar. It is having 2 monosaccharides, glucose and fructose, held by alpha beta 1 to 2 glycosidic bond. So what is alpha? Alpha represents alpha glucose, beta represents beta fructose. Okay. Cellulose. Cellulose is having glucose attached by beta 1 to 4 glycosidic bonds. Trehalose has glucose attached by alpha 1 to 1 glycosidic bonds. Isomaltose is just isomer of a regular maltose. It is having glucose alpha 1 to 6 glycosidic bond which is attached to another glucose molecule. Okay, so all these represent the glycosidic bonds. Okay, alpha 1 to 4 glycosidic bond, beta 1 to 4 glycosidic bond, alpha beta 1 to 2 glycosidic bond, beta 1 to 4, alpha 1 to 1, alpha 1 to 6 glycosidic bond. In this way we can name the glycosidic bonds depending upon what type of sugars are involved and which carbons are involved in glycosidic bond formation. Understood, students? Come on, copy down this. So in your NEET examination you have to memorize maltose, lactose and sucrose. These 3 disaccharides are not required for your NEET examination. Okay, come on. Students, now let us discuss in detail about polysaccharides. What are polysaccharides? Polysaccharides are carbohydrates which are made up of many monosaccharides. There will be several thousands of monosaccharides which will be attached one to another by several glycosidic bonds, which results in formation of long chains of monosaccharides. We call them as polysaccharides, and they may be branched or they may be unbranched. Some of the polysaccharides have branched structure, some may have a linear structure without any branch, and they are less soluble or insoluble in water, mainly because of their large size, and they are not sweet to taste. That's why they are also called as non-sugars, and they does not form crystals. Okay, so polysaccharides also called as glycans. They are also called as glycans. So if you observe the structure of any type of polysaccharide, you can see individual units called as monomers. Individual units called as monomers. You can see here, this is a monomer, this is a monomer. All these rounds represent monomers. These monomers can be any type of monosaccharide. It can be a glucose, it can be fructose, or any type of monosaccharide. One monomer is attached to another monomer by a bond called glycosidic bond. The first is attached to second is attached to third, again by glycosidic bond. This process repeats again and again. This is called condensation or polymerization. Okay, so several monomers are attached end to end to form a long chain by process called polymerization, also called condensation. During this polymerization process, water molecules will be removed. So what is the bond which is formed between the monomers? It is glycosidic bond. Okay, several monomers will be held with each other by bonds called as glycosidic bonds. This results in a long chain of long chain of monomers, we call it as polysaccharide. So I will show you with a model which I have prepared. Look here students, if you assume that this plastic ball is a monomer, a monosaccharide, it will be forming a bond with the second monomer. So the bond is glycosidic bond. The second will form another glycosidic bond with third. The third monomer will form bond with fourth, fourth with fifth, fifth with sixth, sixth with seventh, and on again. So because of this repeated process called polymerization, a long chain of monomers is formed, which is called what students? It is called polysaccharide. In a polysaccharide, there will be left end and right end. The left end is called non-reducing end. This end is called non-reducing end, and the right end is called as reducing end. So this is the reducing end, this end, and this end is non-reducing end of our polysaccharide. Understood the concept, students? Understood? Okay, now let us further discuss. What are the functions of polysaccharides? In living organisms, polysaccharides, they carry out several functions, such as they act as reserve food materials. So the excess food material, whatever is present in the body, that will be stored inside the cells in the form of reserve food materials. This is one of the important functions of polysaccharides. And second function, they act as structural materials. They provide mechanical support to the cells. In plants, it is in the form of cellulose. In fungus, it is in the form of chitin. Next, slimes. Polysaccharides can form slimes, sticky material which is having lubricating effect. Okay, and they can also form anticoagulant materials like heparin, which will prevent clotting of the blood. So like this, there are several roles of polysaccharides in living organisms. So first, let us discuss about homopolysaccharides. Students, what are homopolysaccharides? Homo means same. Those polysaccharides which are made up of a single type of monomer formed by condensation reaction, we call them as homopolysaccharides. Homopolysaccharides are complex carbohydrates formed by repeated condensation, or polymerization of only one type of monosaccharide. There will be only one type of monosaccharide which will be repeating again and again throughout the chain of polysaccharide. We call them as homopolysaccharides. Examples: starch, glycogen, inulin. So these three polysaccharides have role of reserve food materials. They act as reserve food materials in living organisms. Starch is present in plants, glycogen is present in animals, inulin is present in plants such as garlic, onion, and artichoke. Okay, next to homopolysaccharides, cellulose and chitin, they are called structural polysaccharides because they have structural role. They form cell wall materials in plants and fungi respectively. Understood? First, let us discuss about starch. What is starch? Starch is a reserve food material of plants. Plants will store their excess food material in the form of starch inside the cells in the form of starch granules. Okay, there will be specialized cell organelles which will store the starch called amyloplasts. These amyloplasts, they store the starch, and the starch will be stored in underground roots and also in underground stem tubers like potato. Plants also store starch in the seeds such as rice, wheat, barley, etc. Okay, so if you observe this starch is playing the role of food material in plants, and it is made up of the monomer called as glucose. Starch is made up of only one type of monomer, that is glucose, which is a hexose sugar, And starch, it is mixture of two polysaccharides, amylose polysaccharide and amylopectin. Look here students, this is the structure of amylose. This is the structure of amylose. It is in the form of a spring, a coiled structure, helical structure. Okay, so this coiled structure is formed by several glucose units which are attached by 1 to 4 alpha 1 to 4 glycosidic bonds. Okay, amylose is a polysaccharide which is component of starch. It is spirally coiled. Okay, so next, attached to this spiral of amylose, we can see several branches of glucose molecules. They are called amylopectin. So these branched chains of glucose which are arising from main helix, they are called as amylopectin chains. So amylopectin will be about 80% in the structure of starch and 20% will be only amylose. In amylopectin structures, which you can see here, several glucose units are arranged in the form of branches. There will be branched chains with alpha 1 to 6 bonds, and there will be also unbranched chains with alpha 1 to 4 glycosidic bonds. So this is the structure of starch. When we test starch with iodine, iodine will occupy the space which is present in the helix. When iodine occupies the space present in the helix, the entire starch will show up in blue color. Okay, so what is the test for starch? Iodine test. The iodine test will give blue color. So when any carbohydrate, when it is tested with iodine and it is giving blue color, it is indication that that unknown carbohydrate is starch. And why starch gives blue color? Because iodine occupies the space present in the helices, in that spring-like coiled structures of amylose. Understood? And starch is present in cereals like rice, wheat, barley, and also in potato. I will show you the real examples of these materials. Look here students. In this plate there is rice. These rice grains contains starch. So when you cook the rice, you get cooked starch. It will have more amount of water. But right now there is very less amount of water in this rice. This is starch, pure form of starch. Okay. Next, another example for starch is potato. So inside the potato, if you open the peel, if you scrape out the peel of potato, you can see inside there is a starch. This is the starch. So if you do iodine test of potato and rice, you will get blue color, indicates that this is a starch. Understood the concept students? Copy down. Students, now let us discuss about another important homopolysaccharide, glycogen. Glycogen is also called animal starch. Why it is called animal starch? It is a very important reserve food material which will be present in animals just like starch in plants. That's why there is another name for glycogen. It is called animal starch. Okay. Now let us see some points about glycogen. It is a storage polysaccharide in animals and also in fungi and also in bacteria. Okay, so it is also called animal starch, just now we have discussed. And in animals, glycogen will be stored in liver and muscles, these two organs. So whenever animals have requirement of energy, at that time the glycogen which is stored in liver and muscles will be simply broken down to produce glucose molecules, and glucose will be use it to produce energy in the body of animals. Understood students? Now, it is a polymer of alpha glucose. Just like starch, our glycogen is also a polymer of alpha glucose monosaccharides. Look at the structure of glycogen students. So this is the structure of glycogen. In glycogen you can see straight chains and also you can see several branches. Okay. So in the straight chain of glycogen, you can see each molecule represents alpha glucose. This is alpha glucose, this is also alpha glucose. All these molecules are alpha glucose molecules. How they are attached with each other? By alpha 1 to 4 linkages, alpha 1 to 4 glycosidic linkages. Okay, so in glycogen structure, the straight chains have alpha 1 to 4 linkages or alpha 1 to 4 glycosidic links. Then there are side branches which are attached to straight chains by a special type of linkage called alpha 1 to 6 linkage. What is alpha 1 to 6? Look here, students. Here, the first carbon of a side branch is interacting with the sixth carbon of a main chain to form a bond called alpha 1 to 6 glycosidic bond. So this bond is called alpha 1 to 6 linkage. Okay, so like this, several branches will be attached to the main chain. You can see here the cartoon diagram of glycogen, which is given in NCERT textbook. This is NCERT diagram of glycogen. The cartoon diagram of glycogen, you can see here, this is the main straight chain of alpha glucose molecules attached to each other by alpha 1 to 4 linkages, and there will be several branches connected to each other by alpha 1 to 6 linkages. Understood, students? Now, starch gives blue color with iodine, but our glycogen, it gives red color with iodine. That is the difference. Understood, students? Now let us discuss another storage polysaccharide in plants, inulin. Just like our starch, inulin, it is also a reserve food material in some plants. It is a homopolymer of a monomer called fructose. Fructose, we have discussed about the structure of fructose. What is the formula of fructose, children? C6H12O6. Just like glucose, fructose also have same formula, but our fructose has a different structure. It is called as furan ring structure. Okay, it is a homopolymer of fructose monomers with beta 2 to 1 glycosidic linkages. Several fructose molecules are interconnected. They are connected to each other to form a very long chain. Okay, we call it as inulin polymer. So look here. Once again, we will take our model. Imagine that each ball is a fructose. I am drawing F here. F for fructose. One fructose is attached to another fructose molecule. Another fructose molecule. By what glycosidic bond? By beta 2 to 1 glycosidic bond. So like this, several fructose molecules are attached to each other. It gives rise to a long chain of fructose molecules. We call this as inulin. Understood? So that is the concept. Now, it is storage polysaccharide in plants like dahlia, onion, garlic and artichoke plants. In dahlia, it will be stored in underground tubers. In onion and garlic, inulin will be present in the bulbs. In artichoke, it will be present in the shoot. So in this way, inulin acts as a reserve food material in some plants. Okay, inulin is not digested in human body. Even though we eat onion, garlic, dahlia tubers, but we cannot digest inulin because there are no enzymes which can digest inulin in our body. Our enzymes cannot break beta 2 to 1 linkages present between fructose molecules. That's why we cannot digest inulin. Okay. Next, it does not show any color change with iodine. So our glycogen, it shows red color, but inulin, it does not show any reaction. There will be no color change even with iodine test. Okay. So this is about inulin. Copy down, students. Now, three important points from NCERT book we have to discuss about storage polysaccharides. Starch, glycogen and inulin, these three are storage polysaccharides. Why living organisms, they prefer to use these storage polysaccharides as reserve food materials? Because there are some reasons. Storage polysaccharides are more convenient storage molecules than any other molecules in the living organisms. Why? Because they are less soluble in water. Storage polysaccharides are less soluble in water because of their large size. So storage polysaccharides, they have very large size and they have high molecular weight, so they are not so easily soluble in water. So that will be a benefit for living cells to store food material. Next, storage polysaccharides does not cause osmotic imbalance in cell. So whenever simple sugars are there, it is very difficult for the cells to maintain them inside the cell because they cause osmotic imbalance. Whenever sugars are present, then water will start coming into the cell because of osmosis. But if storage polysaccharides are stored inside the cell, there will be no problem of osmotic imbalance. So that is another plus point for cells to use storage polysaccharides as reserve food materials. Next, another benefit of storage polysaccharides is that storage polysaccharides occupy small space because they are compactly folded. So inside the living cells, our storage polysaccharides get compactly folded and they occupy only little space inside the cell. So this is another advantage for the cells. So that is the reason why, these are the three important reasons why the living cells or living organisms prefer to store their food material in the form of the storage polysaccharides like starch, glycogen and inulin. So question definitely will come from these 3 points in the NEET examination, students. Note down. Now let us discuss about structural polysaccharides. Structural polysaccharides are also a kind of homopolysaccharides. They have structural role in living organisms. They provide mechanical support to the cells. Okay. The two structural polysaccharides are cellulose and chitin. Cellulose. Cellulose is a homopolymer of beta glucose monomers. That means beta glucose monosaccharides are held to each other by several glycosidic bonds to form a very long chain that results in formation of cellulose. So it is a homopolymer of beta glucose monomers held by beta 1 to 4 linkages. Look here, this is a beta glucose molecule. One beta glucose molecule is attached to another beta glucose molecule by a linkage called beta 1 to 4 linkage. So like this, the process repeats again and again between the beta glucose molecules to form a long chain which results in formation of our cellulose polysaccharide. Understood, students? So look here, this is the model. By using this we can understand the structure of cellulose. Now each ball represents beta glucose. So two beta glucose molecules are attached by beta 1 to 4 linkage. Again, next beta glucose, one more beta 1 to 4 linkage. This process repeats again and again to form a long chain of polymer which is called cellulose fibril. So like this, cellulose is formed. If you break these bonds, if you break this beta 1 to 4 bonds, so what monosaccharides you will get? Obviously we will get beta glucose molecules. Okay, so now let's see other points. It is present in plant primary cell wall material. So inside the plants, there will be plant cells. The plant cells, the primary cell wall, this is called primary cell wall. This primary cell wall will be made out of our cellulose, and it is also present in cotton fibers. So cotton is the purest form of cellulose. Look here, students, I have brought some cotton for you. Yeah, this is the cotton. Okay, if I pull the wick, I can extract cotton fibers. So these cotton fibers contain purest form of cellulose. Understood? So that is about cellulose. So cellulose does not produce any color with iodine. So iodine test, cellulose does not respond to iodine test. It will not produce any color, and it is not digestible by humans. Human beings, we cannot digest cellulose. Even if you eat cotton, we cannot digest cellulose because our enzymes cannot break beta 1 to 4 linkages present between beta glucose molecules. So the enzymes present in a human body, amylase is the enzyme. What is the enzyme? Amylase. Note down the students. Amylase is produced in salivary glands and also by pancreas. The amylase enzyme, it can break only alpha 1 to 4 glycosidic bonds, okay, which is present in starch but not in cellulose. Understood? So that is the point. Amylase can only break alpha 1 to 4 glycosidic linkages present in the starch. That is the reason only starch is digested in human body but not cellulose. Got the point? Now, chitin, next structural polysaccharide, chitin. It is again homopolymer of a special type of sugar called N-acetyl glucosamine, NAG. NAG stands for N-acetyl glucosamine. So one N-acetyl glucosamine sugar is attached to another N-acetyl glucosamine monomer by a bond. The name of the bond is beta 1 to 4 bond. So this repetition of this bonding, it will result in formation of a long chain of polymer which gives rise to chitin. Chitin, it is present in cell wall of fungi and it also forms exoskeleton of insects. It also forms exoskeleton of insects, means what? The outer protective covering which is seen in the insects. Okay, note down this, students. Students, now we are going to discuss the last topic in carbohydrates, that is heteropolysaccharides. Heteropolysaccharides are those polysaccharides which are formed by condensation of more than one type of monosaccharides. Okay, now let us see some examples. Peptidoglycan. Peptidoglycan. Peptidoglycan, it is a cell wall material present in bacteria. So look here, students, this is a bacterial cell. The bacterial cell has the outer protective covering called cell wall. So this cell wall will be made out of material called peptidoglycan, which is a heteropolysaccharide. So it is a heteropolysaccharide of two monosaccharides, N-acetyl glucosamine and N-acetyl muramic acid. Okay, so these two units will be repeating again and again in the structure of peptidoglycan. Next, another example is mucopolysaccharides. Mucopolysaccharides are gelatinous carbohydrates. They have some slimy nature. Okay, let us see examples of mucopolysaccharides. Mucilage. Mucilage is a slimy material which we can see in the case of snails. When snails are moving, they will leave a trail of mucilage, a sticky material. Okay, and mucilage we can also see in aloe vera, students. I have brought for you aloe vera. Look here, a nice beautiful aloe vera leaf. Look here, this is a fleshy leaf. So if I cut open aloe vera, if I cut open aloe vera, then I can see some slime. I can see some slime. Look here. Yes, I will put here. Yeah, let me cut it open. Can you see the strands of slime? Yes, so this is the slime. This is the slime. This is the slime. So this is mucilage, students. This is a gelatinous mucilage. Okay, so this can be called as a mucopolysaccharide. It is also a carbohydrate. Look here. Understood? Understood? Yes. Let me clean my hand. Yeah. So it is slimy to touch. So now let us continue. Yes, that is about slime. Next is phycocolloids. Phycocolloids are slimy material which we can see in the algae such as agar and algin. They protect the outer surface of algae from desiccation. That is the function of phycocolloids. So phyco represents algae. Colloids means colloidal material, jelly-like material which forms the outer covering of algal bodies. Okay, next is hyaluronic acid. It is another heteropolysaccharide which is mainly seen in animals in synovial fluid of joints. Okay, so this hyaluronic acid, it gives a lubricating effect for our joints to move freely. Okay, it is also present abundantly in our knee joint, hyaluronic acid. Okay, and it is also present in our eyeball. Okay, next is heparin. Heparin is an anticoagulant. It is present in blood. It prevents clotting of blood or coagulation of blood. So this is about different types of mucopolysaccharides. Understood, students? Next, the last and but not least, we are going to discuss about reducing sugars and non-reducing sugars. What are reducing sugars? Those sugars which can act as reducing agents are called as reducing sugars. All monosaccharides, your trioses, tetroses, pentoses, hexoses, heptoses, all monosaccharides act as reducing agents. They are reducing sugars. And our disaccharide, maltose, lactose, cellobiose also act as reducing agents. They are reducing sugars. Next, non-reducing sugars. Non-reducing sugars are those sugars which does not have reducing activity. They cannot act as reducing agents. Example, sucrose and trehalose. Sucrose is our cane sugar and trehalose, it is called insect sugar. It is present in insects. So this is about carbohydrates, students. We have completed carbohydrates. Let us celebrate. We will again join in another class. We will start another biomolecule. So keep watching. Share this video with your friends. All the best. Keep preparing for your NEET examination.